Good morning, and Professor Nihal Thomas and Dr. Jacob John, others who are here, and those who are online. It's really a privilege to be here this morning, and I'd first like to really appreciate CSCR and Professor Nihal Thomas for setting up this series of talks, which have really highlighted a lot of the research that's been happening over the years and the lessons that we have learned from that. Thank you, Dr. Nihal. Today, it's my privilege to introduce Dr. Gagandeep Kang. Dr. Gagandeep Kang is no stranger to any of us. She's been a scientist, researcher, and a public health physician, as I would put her primary role as. She has really been someone who's led the institution in research, be it as a vice principal for research or later as a mentor for a whole lot of us in the institution. Dr. Kang is at the moment the director of Enterics, Diagnostics, Genomics, and Epidemiology at the Gates Foundation. She has been instrumental in transforming infectious disease research in our country. And as a result of which, she has had several honors that we are aware about. She was the first woman Indian scientist who's been a fellow of the Royal Society in its 360 years history. She's also been she's been awarded the Infosys Award, being in biomedical space. She was the first person to receive it. She's got several other awards that have come up. I'm not going to waste her time and ours talking about each one of those, but I just like to specifically talk about some other things about Dr. Kang. As she's an eternal optimist, and she has looked at situations and seen it from a perspective that lots of other people don't. And you will see today, and as part of her lecture, that she's seen these problems and challenges that we face with oral vaccines, protecting children, with a very different light. And she believes that with the right amount of effort and persistence, we'll be able to reach goals and answers to complex problems. And her work over the last 20 years has clearly demonstrated that it's possible. But it took a lot of work. And as you would see today, and I'm sure she'll be talking about it, she built cohorts as a microbiologist and then worked with these communities, understood the problem, saw paradoxes that needed to be addressed, worked relentlessly at it. And in all of that, her primary contribution, she believes, is in mentoring others, in being able to leave for the next generation. And I have spoken to several of her doctoral students and people who worked with her, and I have had the privilege of sitting on the ringside watching what she's done. And it's truly been a privilege to know her, and we're very grateful, ma'am, for being here with us today. Over to you, please.
Yeah, CMC is known for overly generous introductions, but thank you very much, Jacob. Thanks a lot, Nihal, for inviting me to speak to the group here and the group online. I think one of the things people talk about, all the things that a person has done. But I think it's very important to understand that any person, any team is nothing without the environment in which they work. And CMC is such an incredibly enabling environment for people to be able to learn, to work without, there are always constraints, but in CMC generally, with support, you can find help around those constraints. So in the next few minutes, what I'm going to be talking to you about is the vaccine for India by India. This is work that other people started in the 1980s that I got engaged in probably about 25 years ago. And it's a story that continues to evolve. So I'm hoping to take you along on that journey with me. Now, this talk is organized in 3 sections. We'll talk about rotavirus disease and vaccine development. We'll talk about the Indian rotavirus vaccine story. and then what that means for the rest of the world. Now, rotavirus is a pathogen. It's a virus which has a wheel shape. It can be seen here on this electron micrograph. You know, it's a round virus. That's why it's called rota or wheel. It's a very pretty looking virus, as I will show you. And it causes acute gastroenteritis in many species. Humans are one species in whom it is an important pathogen, but you also see it in a number of animal species. So some of the strains we have come from monkeys, come from horses, come from rats. And in every species, when rotavirus causes an infection, the young of that species develop a severe diarrhea. Now, the fact that it caused diarrhea was recognized over a century ago, where it was called duovirus because it had a double-layered structure on pictures like this electron micrograph. The fact that it caused diarrhea in humans was only discovered in 1973. The lady there is Ruth Bishop. She is the one who studied biopsies under the electron microscope with a couple of her colleagues, one of whom wound up coming to Vellore, Ian Holmes. And they were able to show that in children who had unexplained severe gastroenteritis, when you took a duodenal biopsy, you could see many, many aggregates of these viruses. So that was a revolutionary discovery because most of the diarrhea in the very young had been unexplained until then. In fact, when I was a student here, we used to think that, you know, in the microbiology lab, you studied bacteria. You almost never studied viruses. And viruses as a cause of diarrhea were investigated only because CMC had an electron microscope. So we had Ruth Bishop, we had Ian Holmes, and in the 1970s, some very prescient people at the Wellcome Trust Research Laboratory, as it now is, which was the Wellcome Research Unit then, wrote to the Australians and said, we have an electron microscope, come and show us how to recognize these viruses. So they came, Dr. Minimathan learned how to identify the viruses, and they were able to show that rotavirus was a cause of gastroenteritis in many outbreaks of gastroenteritis in India, as well as in endemic disease in India. So the first publications from CMC on rotavirus started in the late 1970s. Now, group A, rotaviruses, there are many groups of viruses, and the virus actually has a structure that is not a double layer as we thought initially, but it is a triple layered particle. And it's made up of 6 structural and 6 non-structural proteins. In those structural proteins, what we call group A viruses, which are the commonest cause of gastroenteritis in children, it is that blue layer of VP6 that determines the grouping of the viruses. And then you have proteins on the surface shown in yellow and red. And it was believed that you needed to make antibodies to those yellow and red proteins in order to be able to get protection from disease. Now, this was a lot of data that was based on animal studies, and it was in the late 1970s that people started to try and make vaccines for this viral infection. Now, you can see the child there with rotavirus. I don't know if you can see that the skin is really quite wrinkly. And this is a typical picture of dehydration, where a child loses fluid very quickly. Now, in all diarrhea, you lose fluid. Adults also lose fluid. But what happens in many viral infections is you get not only diarrhea, you also get vomiting. So if you're losing fluid at both ends, then dehydration can happen very quickly. and children can die if they don't receive attention. Now, if you can rehydrate a child with oral rehydration solution, that is fantastic. It's really, really good. It can treat mild infections very easily. It can even treat severe infections if you get oral rehydration initiated early enough. But frequently, what tends to happen is that children dehydrate very quickly at home, and then you need to get them to a facility to try and rehydrate them. Very difficult to start an IV line on a dehydrated child. So if you don't have skilled people there, then children can die. If they come to CMC, there is absolutely no reason for any child to die of a diarrheal illness. But you can imagine that if they are in a rural facility somewhere where you don't have someone capable of starting IVs on very small babies, then it can be a challenge. There are in mission hospitals people who rehydrate by putting needles into bones, into the scalp, into feet. That can work, but not always. So rotavirus can kill if children cannot get to care. And the reason that people began to focus on vaccination as a tool to prevent rotavirus is because vaccination is predictable. Disease, infection, and disease are not predictable. So in vaccination, you can bring the child for immunization. If a child gets sick at home, what is it going to take for you to bring that child to care? You can't decide that and arrange for that ahead of time in places that have poor healthcare system. So that's why people began to focus on prevention, even though we know that rehydration is an effective treatment for diarrhea. So when it came to the development of rotavirus vaccines, there were a lot of approaches that were taken. Many of them started by this person, Al Kapikian, who was at the National Institutes of Health. The idea was, can we take a human virus and attenuate it and give that to babies? Can we take animal viruses, combine them with human viruses, create a chimera, and make that into a vaccine? Can we take just straight up animal viruses and put them into human beings and they will induce an immune response that is protective? So multiple approaches that were taken. Most of the strains came from cows and from monkeys. And the very first vaccine was licensed in 1998. This was a vaccine called RotaShield. Now, I'd like to take 1 minute to show you what the world was like at the time that the first vaccine was introduced. This is what the world looked like. So if it's red, that's bad. Light blue is in the middle. Dark blue is there isn't so much of a problem with diarrheal deaths due to rotavirus. Is that clear? Where are most of the rotavirus deaths occurring? In Africa? Yeah, little red dots in Asia. Myanmar is yellow. The US, you can't get darker blue than that. Which was the first country to introduce the vaccine? The US. Why? Why, when your children do not die of this disease, do you introduce a vaccine there? Well, if any of you have had children in your families who get sick, you know what it means for the parents. They have to be looking after the child. So it's not just a question of preventing death, it's also providing care for the child. And in people who did economic analyses at the time, they found that the amount of time that parents and caregivers had to take off from work in order to look after children with rotavirus, it actually meant that even for rich countries like the US or Australia, it made sense to introduce the vaccine to prevent the severe disease that you get due to rotavirus. So care is good. Children would not die, but the illness is still severe enough and common enough that it makes sense for the country's public health program to introduce these vaccines. So in 1998, the US is the first country to introduce the vaccine. This is Rotashield, and they give out about 1.5 million doses of the vaccine, and they find that there is a side effect. The US has a system of reporting adverse events due to vaccination, which is a passive reporting system. And this vaccine adverse event reporting system identified a possible association with a condition called intussusception, a telescoping of one part of the intestine into another. Now, if you look at the figures that are up there on the right-hand side as you face the screen, what you can see there is where it says first dose, you can see that all the cases of intussusception are happening shortly after vaccination. So after the first dose, intussusception cases go up about 1 to 2 weeks later. After the second dose, little bit of a signal. After the third dose, nothing. Now, intussusception is a very common condition that occurs in young children in any case. It happens without vaccination. But after vaccination, you saw this clustering, and that meant that there was likely to be an association that vaccine was resulting in increased cases of intussusception. So this was investigated, and subsequently, any studies that were done for rotavirus had to be big enough to evaluate the risk of intussusception or decide that they were not going to look at that risk. Initially, it was believed that intussusception, you had to vaccinate 4,000 children to get one case of intussusception that was vaccine associated. Later, they figured out that it was about 11,000 children needed to be vaccinated for one case of intussusception. Now, again, intussusception is a common surgical emergency. Nowadays, management of intussusception has advanced so much that it doesn't go to surgery. Surgeons might oversee cases of intussusception, but most treatment is just giving a child an enema. And those can be air or water enemas, and those reduce the intussusception quite easily. Nonetheless, this was almost 30 years ago, and intussusception was a concern. The vaccine was withdrawn. But two companies continued to work on rotavirus vaccines, and they licensed two vaccines in 2006. They ran very large trials, 60 to 70,000 children. These were designed to assess a risk of intussusception similar to Rotashield. I'll give away the next slide and tell you they found no intussusception in the trials. And they actually brought the vaccines to market. The data were published in the New England Journal of Medicine in January of 2006. The vaccines were licensed and were introduced almost immediately into the programs in the US and in many high-income countries. The cost of the vaccines at that time was about $200 to vaccinate one child. Now, in India, with 25 million, at that time, 27 million children, we did a back of the envelope calculation, and we found that if you took India's entire health budget and you wanted to vaccinate children with this vaccine, one vaccine against one disease, at this cost, it would be 40% of India's entire health budget. So include the AIMS, include every public hospital, include every service that we have. It was 40% of all of that to vaccinate children with one vaccine. So obviously, this was not something that India could afford to do. Now, these vaccines became introduced into India in the private market, and there they cost either 1,800 or 1,200 a dose, still an incredibly expensive vaccine. Now, when these vaccines had to be licensed, studies had to be done in the 60 to 70,000 participants. And what I'd like to show you here is the data from the two vaccines. Basically, if you looked at the Rotarix vaccine, it had 85% efficacy against hospitalized rotavirus gastroenteritis. Rotatec had 95% efficacy. Rotarix was a human-based vaccine, single strain, and you needed to give 2 doses. And Rotarix was a combination cow virus, human virus vaccine, and that had 5 different strains in it, and you needed to give 3 doses of the vaccine. So that was vaccine development until 2006. Now, if we think about the Indian rotavirus story, we start first from, is rotavirus a problem in India? Now, I told you that Dr. Minimathan had started studying rotavirus in the Wellcome Research Unit in the 1970s. In the 1980s, she took part in a very large WHO study that was across many countries looking at causes of diarrhea. And what they found there was that there were 4 pathogens that were common causes of diarrhea around the world, and rotavirus was one of those 4. It was about 20% of all diarrhea anywhere in a low and middle income country was due to rotavirus. Now, these studies were done in the 1980s, and people didn't really have much of a focus on diarrhea after that. But there had been work that had been continuing that was being done by the person whose plaque is outside this room, Dr. M.K. Bhan, who was studying diarrhea in networks, mainly in North India. And he was finding a lot of rotavirus, and he was thinking about developing a vaccine. So in 2001, Dr. Bhan called a lot of us up to Delhi and said, you need to work with ICMR and you need to set up rotavirus surveillance across the country. Given the way things work in India, the discussion was in 2001, the work started in 2005. It took a lot of effort to get it across the line, but ultimately we were able to develop a network of laboratories, a network of sites where we could do surveillance across the country. In the first phase, we had only 11 places. In the next phase, we wound up with 38 sites that were doing surveillance for rotavirus. What we found in those studies was that our 20% figure had changed about 37%. So 2 out of every 5 children hospitalized for diarrhea in India actually had rotavirus. So these were findings that told us that rotavirus was important. And in work that Jacob did, collating data from multiple sites across the country, work that we had been doing in the community over here with JP, we were able to come up with what is called a disease pyramid for India. And this is based on the 2011 birth cohort of approximately 27 million children in India. The big numbers are on this side of the pyramid, but I'd like you to focus on the other side of the pyramid in terms of what it means for a child that is born in India. So 1 out of every 2 children born in 2011 would have a rotavirus gastroenteritis. 1 in 8 children would need an outpatient visit. 1 in approximately 30 children would be hospitalized, and 1 in 650 children would die of a rotavirus gastroenteritis. This is how important this very neglected area of medicine is. So I hope that with this figure, I have convinced you that in India, just like in the US and Australia, where their worry was mostly money, our worry was actually disease. We had so much disease that we needed to think about having a vaccine. So, we were pretty convinced we needed the vaccine. The question was, which vaccine? And will it work in India? We knew from the polio story that oral vaccines don't work very well in Indian children. And then could we afford it? So Rotashield was produced under a license from the National Institutes of Health by Wyeth. But the National Institutes of Health and Dr. Kapikian, as I showed you, had many other vaccines as well. And they will come back into the story later. But at that time, India and the US had an Indo-US vaccine action plan, and that began to support rotavirus vaccines. Now, this was meetings that, so this is the person who was responsible for India's first rotavirus vaccine. Dr. M.K. Bhan was a pediatrician at AIIMS. Can I talk about alcohol in this room? Okay. So Dr. Bhan met a guy called Roger Glass, who was posted in Dhaka. And the two of them were at an Asian meeting on diarrheal disease. And after the day's meeting was done, they retreated to a bar where they had very interesting conversations, and that resulted in a collaboration that lasted over 40 years. So essentially what Dr. Bhan had done is that in 1985, WHO, in this study that Minimathan was also involved in, had made these ELISA tests and distributed them to many labs across the world. AIIMS was one of the places that got the ELISA tests, and Dr. Bhan was looking for a way to get stool samples quickly in order to be able to use those ELISAs. And he thought that neonates would be a negative control. They're too young, they haven't been infected as yet. And then he thought there was something wrong with the test, because he found that a lot of neonates actually had rotavirus. And he couldn't figure it out because these were asymptomatic. So 50% of neonates who had been in hospital for 3 days, and 75% of those who were in the hospital for a week had rotavirus. So one, he found asymptomatic infections of rotavirus, and then he tried to characterize that strain working with virologists. But more important than that, he followed up these children who had had these neonatal infections, and he found that these kids that had the asymptomatic neonatal infection were actually protected against severe rotavirus gastroenteritis later. So this made this particular strain that had been in the neonatal nursery a potential vaccine. So under the Indo-US Vaccine Action Program, getting inputs from Roger Glass, who had access to lots of virologists at CDC, he sent people to CDC to train and managed then to do a lot of work on strain characterization in the US, in India. After they characterized the strain, they made pilot lots, they did phase 1 studies in US adults and US children. And in 1999, they identified a manufacturer who was willing to take on the risk of making a brand new vaccine in India. Remember, 1999 is the year after Rotashield went into the US and is withdrawn. So now there is no rotavirus vaccine. There is the danger of having an unsafe vaccine. And Bharat Biotech says that with the right kind of government support, we are willing to try to make this vaccine. So then they go ahead, they make the vaccine, They do early stage testing in Delhi, and then they have to do a phase 3 study. Now, a phase 3 study is a clinical efficacy study where you need to understand, does the vaccine actually protect against diarrhea or not? And this is the time when I get a call from Dr. Bhan saying, will you take part in the phase 3 study? So 3 study sites are chosen, Delhi, Pune, and Vellore. And then comes the question of, can we really do this study the way we have decided to do it? There is a safety issue. And also, the best way of doing a vaccine study is to have a randomization and blinding. So you don't know who has gotten the vaccine and who has not, and you're still able to look at the outcome. So how does a vaccine study work? You have some children who will get the vaccine, some children who will not get a vaccine. The investigators don't know who it is. You follow them up. You look to see which child has got diarrhea. If the vaccine works, then fewer vaccinated children will get rotavirus diarrhea than children who received a placebo. But the question of ethics then comes up, which is, if you have licensed vaccines in the country, remember Rotarix and Rotatech were licensed in India in 2008, can you do a placebo-controlled trial? So there had to be a discussion with WHO coordinating it, thinking about the ethics of doing a placebo-controlled study in this setting. And what that consultation determined was that placebo controls could be acceptable in certain kinds of situations while you're developing a locally affordable vaccine, while you're testing local safety and efficacy, while you're testing a new vaccine where an existing vaccine is not appropriate, and when determining the local burden of disease. So for us, the first criterion and the third criterion were met, and we were allowed to go ahead with the study. But we were also told that for 2 years, we had to provide all the care that these children needed. So I think our study was the first one. Jacob ran the study, and it was the first one to give cell phones to every study household, to monitor everything about the children, paying from, we paid for hernia surgeries, we paid for open heart surgeries. We looked after children no matter what their needs were. So you had people in each place that were on call 24/7. We ran the study very efficiently, very little loss to follow up, and we were able to produce results in a very short time frame, almost ahead of schedule. And we showed that the vaccine had 55% protective efficacy. Now, this was a fantastic result, but you remember I told you Rotarix, Rotavac were 85 and 95%? The Indian vaccine is only giving 55%. Terrible. Anyway, the vaccine got licensed by the Indian regulatory authorities in 2014, and in the public markets, it was priced at $1 a dose. That meant $3 for a full course. Compare that to $200 for the US markets when the other vaccines came. Now, we had been working, as Jacob mentioned, on cohort studies. And it turned out that our cohort studies were actually very well positioned to explain this 55% efficacy that we were seeing in India. Now, what had we been doing in the cohorts? We worked in Chinnalapuram, Ramnaikapalayam, and Kasba. And essentially what we showed was that in Indian children, if you look at protection against moderate to severe diarrhea due to rotavirus, you need to have 2 or 3 exposures to the virus. These studies were about natural infection, But our data showed that if you had 2 infections, you got 57% protection against moderate to severe diarrhea. 3 infections, you got 80% protection against moderate to severe diarrhea. Now, this was very different from data that had come from other parts of the world. I'm not sure you can see this figure very quickly, but within the red circle are the numbers 87 and 100. That is the protection that babies in Mexico got with natural infection. If they got 1 infection, they got 87%. If they got 2 infections, they got 100%, which means they never had a 3rd infection. In our case, you got just above 50% with 2 infections and 79 or 80% with 3 infections. So no perfect protection in Indian babies, very different from data that was produced in Mexican babies. So there were a number of other features about the study, but we could take these data and plug them into a model and show that if the pattern of infection and protection from disease was what we had seen in Vellore, then the vaccine would have only about 50% efficacy. And it wound up having 55% efficacy. Now, I know this sounds terrible, sounds awful, that children are not being protected exactly where they need to be protected, right? The US burden doesn't kill children. Our burden kills children. We need these vaccines, but the vaccines are not working very well. So what does this mean? So when you look at lower efficacy in developing countries, let's take Malawi and South Africa, right? So this is studies with the GSK vaccine. And what you see is that the vaccine had 50% efficacy, orange box below, 50% efficacy in Malawi, 77% efficacy in South Africa. So vaccine is working better in South Africa? Okay. Now let's look at the orange bar, which is how much disease is there in Malawi? How much do you see of rotavirus diarrhea? So if you follow up 100 children in Malawi for 1 year, 13 of them will get rotavirus gastroenteritis. If you follow up 100 children in South Africa, 5.4% of them will get rotavirus gastroenteritis. So then if you look at a 50% efficacy, in Malawi, you are preventing 6.7 episodes. You started with a high burden, so a 50% reduction still means reducing a lot of disease. In South Africa, you start with a lower burden, and the vaccine works very well. But the number of episodes of illness that you're preventing is lower than in Malawi. So this is just to illustrate that even if a vaccine is not perfect, it still has a lot of value for public health programs. So where were we now? We have shown you that there is disease burden in India. We have tested a vaccine in India, made in India, and the vaccine is working. But now you have to convert this into policy. So what does it mean to convert a product into something that can actually have public health impact? And in India, we have the National Technical Advisory Group on Immunization, and they ask these questions. Is there a significant disease burden? Is the disease a priority for the National Health Plan, for the medical community, for the public? Is it recommended by WHO? Would use of the vaccine promote equity? So disease burden I showed you. Is the disease a priority? Well, it depends. You know, at one time when we were having these discussions, a lot of pediatricians said, we do not need the rotavirus vaccine in India. Why? Because you lose a certain income stream. You have children that you are already treating. This is not a fatal illness if they can access healthcare. So you're going to lose some money if you treat children. The second thing is pediatricians also make a lot of money from immunizations, right? So if you immunize in the private sector, you make some money on that. But if the vaccine is available in the public sector, then that part of your income stream might go away. So initially, there was a lot of resistance from pediatricians saying, don't introduce this vaccine. But if you think about it, this was countered actually by doing a lot of cost effectiveness studies to demonstrate that this vaccine was a cost effective intervention for India. The vaccine had been recommended by WHO. And then the question of, does the vaccine promote equity? Now, in 2008, the vaccine has come into the program. The Indian Academy of Pediatrics recommended the use of the vaccine, first conditionally, but then an unconditional recommendation that children who come to pediatricians should get the vaccine. So that meant that anybody who could afford to pay for the vaccine could get the vaccine. But those who could not afford to pay for the vaccine were not getting the vaccine. So when you put a vaccine in the national immunization program, you are actually enabling the access of poor people to these preventive interventions. So it does promote equity. So use was recommended and accepted in 2015. 2014 was when the vaccine was licensed, and TAGI made a decision in 2015, and then in 2016, the vaccine started to be introduced. Initially in 4 states, then in 2017, an additional 4 states. In 2018, UP, which is bigger than many countries, and then in 2019, we had nationwide introduction of the vaccine. Now, for one vaccine company to provide enough vaccines for all the children in India can be quite a challenge. So it's useful always to have a diversity of suppliers, more than one supplier. And then we had the second Indian rotavirus vaccine. I told you about alcopicyan. This was a vaccine that was made at the NIH. It was a bovine human reassortant. And this vaccine was evaluated in India by the Serum Institute and its partners, also supported by the Department of Biotechnology. And this vaccine showed a vaccine efficacy of about 42%. So against very severe gastroenteritis, about 55%. But this same vaccine was also being evaluated in Africa by Doctors Without Borders. And they found that in Niger, which is a much poorer country than India, the vaccine actually had 67% efficacy. So some variability in efficacy can be because of study designs and the way follow-up of children works. But what we had then was 2 Indian vaccines that were both working. Now, the question always is, when you have this variability in efficacy, this is in vaccine trials, which are very, very closely monitored and conducted. What happens when you roll the vaccines out into the real world? And we had support studies led by Venkat that looked at the impact of rotavirus vaccine after introduction into the universal immunization program in India. Now, if you look at the blue or black lines, that is showing you your rotavirus positivity, the percentage of rotavirus positives. And what you can see there is rotavirus is a seasonal disease. It tends to happen much more in the winter, decreases in the summer. And what you can see is in these hills and valleys, once you've introduced the vaccine, the orange line, the hills become less high, right? So you follow this over time, and the longer the vaccine is in the program, the lower the rotavirus positivity becomes. So clearly, the vaccine is working. As coverage goes up, rotavirus positivity comes down. And remember intussusception? So in the story I've told you, I didn't talk about all the problems that we had during the conduct of the vaccine with people attacking us because they said we weren't monitoring intussusception or we were monitoring intussusception too well. So much of the data of the vaccine trial actually wound up being part of litigation that was brought against the Department of Biotechnology and CMC about intussusception. So instead of responding in the courts, we thought we would respond with data, and that's what we did. We essentially showed that there was no risk of intussusception. So you remember I showed you after the first week of the first dose, there was a peak in intussusception. The peak was a little smaller after the second dose with the RotaShield vaccine. These are data from RotaVac. And as you can see, in the first week after the first dose, there is no signal. After the second dose, no signal. After the third dose, no signal. So we've shown that the vaccine works in the real world, that the vaccine is safe. So what does this mean for everything that we learned on this really long journey? Well, it takes a really long time, and it takes a very large team. Within the institution, outside the institution, it takes a lot of effort, and problem solving comes with experience. Every time you encounter something new that is a challenge to you, it sounds very cliched, but it is an opportunity to grow, to learn how to handle things much better. The next time around will be easier. I'll just show you very quickly. When vaccines were introduced in the UK and India, so if you can take a look at the measles vaccine, for example, introduced in the UK in 1968, in India in 1986. After measles, India doesn't really introduce any new vaccines into the national immunization program for a really long time. So I'm just showing you this to tell you that there is a big gap between when a vaccine is introduced in the West and when it is introduced in India. So 68, 86, 92, 2009, 2006 pneumococcal disease to 2020 in India. These big gaps. What is the world doing about addressing these gaps? Well, we have an organization called Gavi, which was founded in 2000, and it was founded to address vaccine inequity. And Gavi's done a lot of work. It's immunized 1 billion children. It's introduced 60 new vaccine programs, et cetera, et cetera, et cetera. You know, they've been strengthening health systems, making sure new vaccines are introduced. But I want to tell you something about what it means when you know systems well. And to illustrate that, I'd like to walk you through this table. What you can see is 3 colors, and you've got lines that are solid lines, and you have lines that are dotted or dashed lines. So the solid lines are when a vaccine goes into a country, a rich country, a country that is not Gavi eligible. And the dotted line is for when the country is a poor country. So let's take the orange line first. The orange line is pneumococcal vaccine. You can see pneumococcal vaccine keeps climbing. Solid line keeps climbing. And if you take the poor countries, what happens is they start much later. They go up slow, but over time, their coverage exceeds that you see in high income countries. If you take a look at rotavirus, same story. Blue line starts in high income countries, big gap with low income countries. But once Gavi gets into the picture and starts providing vaccines to poor countries, coverage in the poor countries is better than coverage in the high-income countries. Now, we don't see that picture for HPV. Why is that? And I'll give you one simple explanation. The PCV and the rotavirus vaccines are made by vaccine manufacturers in India. HPV, as yet, is not being provided to the Gavi markets by a developing country vaccine manufacturer. So when we do something for India, because our population is large, we really need to deliver accessible, affordable, high quality vaccines. When we do it for ourselves, we actually do it for the world. So I had shown you the picture of what diarrheal deaths were like in 1998. I'm going to take you now to 1990 and then a comparison with 2019. As you can see, the picture has changed. The Indian vaccines, the Gavi market started to use vaccines from GSK and Merck, but GSK and Merck always had limits on how much vaccine they would give to Gavi. Most of their vaccines went to high-income country markets because those have a much bigger margin, and they provided a small amount of vaccine to Gavi to provide to countries. But even with that, you can see that diarrheal diseases have come down over time and vaccines have been introduced. Now, this is a map of the world showing rotavirus vaccine introduction. The green countries are the ones that have introduced vaccines. The yellows are the ones that are planning to introduce vaccines. And in 2018, the 2 Indian vaccines became pre-qualified. That meant UNICEF could now buy them for the Gavi market. And in 2023, 3/4 of the vaccines that went to developing countries through Gavi were actually made by Indian manufacturers. So as I said, solving for ourselves actually leads to solving for the world. But stories don't end there, because you think you've done something wonderful in 2023, here we are. And then in 2024, you have to keep your WHO pre-qualification. They got it in 2018, they had to keep it, and then WHO planned to change the rules. So they wanted the vaccine to be sterile. Now, this is an oral vaccine, right? It's going into an environment that is very contaminated. So previously, what was acceptable was aseptic manufacture. So you manufacture in controlled conditions, and then you develop the vaccine so that it is under conditions where contamination is very unlikely. If you introduce a requirement for the final product to be sterile filtered, that means you will lose most of the virus in the vaccine. So to be able to make the same number of doses would require a much larger production, which means your prices would then go up. So then you have to do a negotiation with WHO's expert committee on biological standardization to make sure that the language enables the vaccines that currently exist to stay in the market. So it requires, a lot of effort to get the vaccines there, but it's not like the effort stops once the vaccines are there. So I'd like to leave you with that, the story of work that has been done largely in India, but leave you with a framing of what work in India really means for the world. And with that, I'd like to thank a gazillion collaborators and friends that have been along for this journey, but mostly the children in the first rotavirus study that I did, which was called the Childhood Rotavirus Infection, or CRI study. These kids have all grown up and were followed up until they reached adulthood. So thank you, and back to you.
Thank you, Dr. Kang, for walking us through over 20 years of history with rotavirus vaccines. Today, we have the entire country covered by the vaccines. I open this out for questions and comments at the moment. If you have any questions, if you're online, you could just raise your hand and we'll get to your questions. For those who are in the room, we have mics here. You can unmute and ask the questions. So if there are anybody who has questions online or comments, please unmute and speak. You're able to do that, I believe.
I have a question to Cherry.
Yes, please go on.
With the recent cutback on funding to Gavi, how is Gavi going to find the resources to meet the increasing need for vaccines in the developing world?
Well, there are going to be two approaches to that. The first one is that Gavi has always been intended to have countries graduate from Gavi. So we started with 78 countries. We are under 50 now. And it is a programmed decrease in support that happens to each country, so that countries take up their own immunization programs for themselves. The second is a prioritization exercise, which is going to be done in consultation with countries to see what they view as their most important priorities. The vaccines that are in the routine childhood immunization schedule will be the ones that will be protected. It will be the campaigns and the new introductions that are most likely to be reduced if funding is not available.
Thank you, Jerry, for that very enlightening talk and how you've managed to implement things from a public policy perspective. I have a relatively naive question. In nature, you have animals, and in the process of man-animal conflict, you have death of certain animals, with which certain other animals may get a survival advantage. If you eradicate one virus from the gut, will you have other viruses which might proliferate and cause a problem?
So I think that is an interesting question, and the way it has played out in the vaccine use field is that you do see some strain replacement in pneumococcal strains, but you don't see one virus taking over the niche of another virus in the gut. It may happen in the future, I don't know. But so far, we have no evidence to show that in countries that have very high levels of coverage with certain kinds of vaccines that have actually seen a huge decline in viral populations, have seen something else come up instead.
This again, thank you, Cherry, for that wonderful talk. My question is again on the lines of what Dr. Marvin asked. We've been seeing a lot of vaccine shortages in the country, and a lot of it is because one of the vaccine manufacturing plants, Mithosoli, has closed. We've not been able to vaccinate any adult against hepatitis B for a while. So do we anticipate that the same thing will happen to childhood immunization vaccines? I think everybody's been concentrating on that. What will Gavi step in to address some of these issues?
I think for India, the more we rely on getting our system stronger, the better off we will be. We are a Gavi graduated country. So even though there are some special provisions always for India, but we have graduated from Gavi, and I think we should learn to rely on ourselves. Our public procurement systems, which are the ones that really matter, do have to have a more efficient system to prevent stockouts of vaccine. It isn't that the supply isn't there, it is or manufacturing capacity isn't there. It's just that the processes of approvals for batches and their release can sometimes take time.
Shri, I have another question. When we started off with this vaccine, and you knew very well that this is going to be something like 50, if you move away from there, that it would be about 50-55% efficacy. But yet, in record time, we were able to get that into the health program. What do you think were the key drivers? What strategies did you employ, the group look at, that actually enable this to happen in time? Because sometimes we have great products, but they don't necessarily translate into things, and typhoid is another story.
Well, I think two things. One, you need champions. So people within government that are willing to say that this is something that is important and needs to be pushed forward. And those can sometimes be quite rare to find in government. And the second approach, which is the long, hard slog approach, which we took, is to make sure that you have plugged every gap in evidence. So no matter what people are likely to ask you, in order to make a policy decision, we made sure that that data was there ahead of time.
Like why the progress only the kids not done.
So like many childhood infections, what happens is that you acquire immunity after repeated exposures. So adults rarely get rotavirus diarrhea. They can, but the times when adults get rotavirus diarrhea is either when they are elderly or if they are exposed to a very high infectious dose of the virus that overcomes the immune response that the person has acquired. In children who have not had prior exposure to rotavirus, once the maternal antibodies wane, which happens in the first 3 or 4 months, you see rotavirus rates start to go up. In the West, rates used to go up till about 5 years of age. In developing countries, because we have greater exposure, it would start early, like 4 to 6 months, and then pretty much be done by the time a child was 2.5 years. As we showed in our natural infection studies, repeated exposures do give you protection. It's not perfect protection, but being infected at least 3 times with rotavirus gave you 80% protection. So there are many features. So, there are people and then there are environments. And the environment can really affect how your gut works. So in developing countries, we have very early exposure to gut pathogens. We see a lot of gut pathogens, much more than you see in the West. And that's exactly the stage at which you give the vaccine. So there are many unanswered questions about why vaccines, particularly oral vaccines, don't work very well in people in developing countries. Most of them relate to damage to the gut or the potential for tolerance to be induced to certain kinds of pathogens. you see something so often that you don't react to it. So you treat the viral vaccine as just something that is passing through the gut, so you have no reaction. The other option is, your gut surface is damaged and therefore you cannot respond to the infection. So multiple reasons. We've tried lots of approaches to see if we can have an intervention that solves that problem. Every study pretty much that we've done has been a failure. So the ultimate answer is make our living standards the same as Western living standards. The vaccines will work better, but we will in any case need them less than.
Raj, please ask away.
Good morning, sir. First of all, thank you for this opportunity to listen to ma'am. I've been fascinated by her always, and today I've got the opportunity. So, ma'am, my question is that what do you think the next 5 to 10 years, kind of what diseases should we target for the vaccinations in coming years?
So there's no shortage of potential pathogens that can be targeted through vaccination. The easiest targets are usually viruses, and then comes bacteria and parasites. So I think what we have now is vaccines have been made for most of the globally prevalent pathogens. Some of the vaccines are not great. So if you were to ask me for priority vaccines, I think we should be targeting a much better influenza vaccine, because that is something that historically has been left unaddressed. And then if we were to think about diseases that are important but continue to be a humongous challenge, I would think TB and HIV would be the next diseases to look at trying to make vaccines.
Thank you.
Sorry, we're not able to read. Yeah, just open the chat. Yeah. Yes. Okay. So, Chiri, you want to?
No, I'm happy to respond to the question about pediatricians as being against vaccines. I don't think pediatricians are against vaccines. It was a question at the time of pediatricians saying that the vaccine did not need to be in the national program because this was a disease that could be handled quite easily without vaccination. And the other counter was, many vaccines, for example, we have now chickenpox vaccines or varicella vaccines. These are not in the national program. And if you deliver an expensive vaccine or you set up a vaccination clinic, it gives you the opportunity to earn some revenue. So it's not to say that anybody is against vaccines. Well, some pediatricians, are against vaccines. They're the ones that have previously come after us. But in general, I don't think pediatricians are against vaccines. It's a question of what's a priority for the program, what's the priority for the child.
There's one more question there. Hi, Padma. Padma, you want to speak up?
Oh, yeah, I can answer Padma's question. Padma, we tried really hard. I think we've done about half a dozen or more clinical trials trying to improve the performance of oral vaccines. We've failed in every endeavor so far. But in my new role at the Gates Foundation, I'm still trying.
Terry, that was a wonderful talk. It's not just about vaccines, but about life in public health, where you just keep at it and get where you have got with the vaccine story. I just ask this because very often in other scenarios as well, sometimes not a good enough response becomes a deterrent to keep at it. So it's really nice that you shared that you tried your best. And if this is what was the best and it was still good enough, then we don't give up. Thanks so much for sharing.
Thanks, Padma.
Great talking. Since this is the first generation whole variant vaccine, so is there any advances in the next stage, like either protected antigen or mRNA based,
Yeah.
so that the efficacy can be improved further?
So the question with mRNA vaccines has been, given their reactogenicity, do you want to give them to really young children? But in terms of looking at nanoparticle or subunit based vaccines, there are ongoing efforts to do that.
Excellent work, ma'am, that you always inspire us, and you have done the work for our country and also the world. I have a very naive question. Like you mentioned initially, they wanted to use as a control the newborns, and you mentioned that there were children who were having this virus, but they were asymptomatic. So why they were asymptomatic even though they have virus?
So that's a really excellent question. And that question was answered many years after the infections were documented. It was to do with the strain that was infecting the children. It was a single strain, which was classified as a G9P11 strain. And that's the strain that ultimately became a vaccine. It was believed that the reason that it was asymptomatic was that the P11 of that strain was actually derived from cows. So it did not infect at a very high level and did not cause diarrhea in human babies.
So then if it was asymptomatic, we can directly use that virus even without inactivating.
That's exactly what we did. We just grew it in and standardized the dosing.
All right, we could keep asking generic questions through the day, but we have to get back to work. So thank you. Please join me in thanking Professor Kang for truly been exceptional work over the many years. And we're so grateful to you as CMC community for what you've done, not just in the research, but showing us that in the face of adversity to keep going. And you've been an inspiration for us. Thank you for joining us today. And thanks everyone for being on. Thank you.